All right, folks, we will go ahead and get started here. We've got about 99 people on the line right now, and I anticipate folks will still be logging in here. My name's Erin Fox. I will be kind of moderating the back end of this conference, so if you need anything, don't hesitate to get in touch. Just a couple of logistical items before we go ahead and get started. I can get the next slide. There we go. Thanks to everybody for joining us in this virtual format. I know this is a little bit different than our typical in-person symposium. We can't all network with each other and chat, but hopefully this is a great alternative in these unique times we have here. Just a couple of housekeeping items. The event evaluation, it was in that initial email that I sent out last night, but it is also in the chat box, the very first message in the chat box. So if you open it up and scroll all the way to the top, the link is there. It asks you to rate each and every one of our presenters, and we do have quite a few presenters this morning. So if you do want to open that up throughout the event, that's kind of how we encourage to do it, just so you don't lose track of which person presents what and whatnot. So, but you're always welcome to fill it out afterwards as well. Just a tip there to keep everything organized. We'll be running this just like a normal Zoom meeting. So if you have any questions throughout, we always encourage you to ask those. Just unmute yourself and ask them, or feel free to plop it in the chat box. I will note about the chat box, you can either send it directly to me or to the entire group. There is an option there. So just keep an eye on that depending on what you hope to chat. But if you do unmute yourself to ask your questions, just remember to mute yourself again afterwards, just to kind of eliminate that background noise there. We are recording this event. You should have kind of gotten a consent recorded when you logged in here, and we'll later plop it on our YouTube page if you're interested in rewatching the event on our YouTube. URL is right here. If you do have a Twitter page and you're interested in live tweeting the event, we put our Twitter handle here as well, and that's encouraged. We love to see that. So any other questions, feel free to let me know or chat me directly. If you have any questions, you can always follow up as well after to my email address, which you all have. But without further ado, I will turn it over to Dr. Hank Paulson, who's the director of our center, to give you a little bit of background about where we're at the center. Hank?
Thank you, Erin, and good morning, everyone. I know this is a very strange and stressful time, and many of you are confronting lots of challenges during COVID shutdown. Some of you may have lost loved ones, as we have in the center through our staff. And I think this is an opportunity for us to remember what we're trying to do to help those who are experiencing cognitive impairment. Normally, we'd be gathering in a room with about 200 people, shouting at each other, sharing ideas, looking at posters. We're not doing that this year. We are instead going to focus our symposium today squarely on the junior investigators, research education component trainees. The keynote speakers that we had chosen for this year's symposium have graciously agreed to come back next year when we will have a full in-person symposium, I think, in Ann Arbor. And so today we're simply focusing on our rec trainees with a little bit of a background about the center. Next slide. Now, many of you may have seen our logo before. It had 3 mittens. It had 3 different university logos, and we have rebranded ourselves. It's exactly the same group of institutions. It's University of Michigan, Michigan State, and Wayne State working together, unified as a single mitten, with the colors shown here fanning across the state, not simply in the lower peninsula, but the upper peninsula as well. We just launched this yesterday. It's a brand new logo. I hope you like it, and thanks to our team for putting it together. I also want to say that our symposium every year has been supported not only by the universities, but also by the University Research Corridor, the URC. And we really have appreciated their support over the years, and we anticipate that will continue in years to come. Next slide. What is the Michigan Alzheimer's Disease Center? Well, it's one of a number of ADRCs across the country that are funded by the NIH, shown here on this map. It's a little bit spotty in terms of its location, strongly on the West Coast, strongly in the Northeast, strongly in the Midwest, and a growing group in the Southeast as well. These centers work together across state lines nationally to really try to advance what we understand about Alzheimer's and related dementias. there are over 42,000 participants from all over the nation that are participating in the database that is collected by these centers together. Next slide. The data from the individuals who we see in our center, and in our center we see almost 500, have collected data on almost 500 research participants in our first cycle of funding, which is the last 4 years. And the data go to the National Alzheimer's Coordinating Center, which is based in Seattle, Washington. The data that is there is massively big, comprehensive. It includes neuropsychological data, includes neuropathological data, includes imaging data. And these data are available to investigators both within the center network as well as outside the center network. So if you are in here today, listening in and not affiliated with our center, one, we welcome you and we would love to have your interaction with us. But two, you don't need to be part of a center to access this data. And I think it's extremely valuable data and becoming more valuable every day as more is collected. Next slide. We have a lot of studies that we support as a center, and this is simply a snapshot of some of the studies that are directly connected to what we do with our participants. The gold square in the middle is our big longitudinal University of Michigan Memory and Aging project. This is where that almost 500 individuals that I mentioned to you are being evaluated on an annual basis. The data are being collected and sent to the ADRC network at the NAC. But there are many different studies doing everything from non-pharmacological brain stimulation to enhance memory, to couples-based analyses, to imaging studies of subjective cognitive impairment. I'm not going to go through each of these. They're available to you, and this gives you some idea of the range of types of studies, particularly involving people that we support. We also support studies that are more mechanistic and that involve model systems as well. Next slide. This is a picture of the center structure. This is a brand new sort of organizational chart of the center. And I say brand new because we've added 2 new cores. The 2 new cores that are part of the center are the biomarker core and imaging core. Aaron is highlighting those right now. These are still under construction, but they will be an integral part of our center as we put in our application for competing renewal this fall. I direct the center with the assistance of Bruno Giordani, and we have a clinical core, a neuropath core, a data core, and an outreach and recruitment core. What I'm going to focus on here, though, is the one at the bottom, which is the research education component, because a vital part of any ADRC, and certainly our ADRC, is that we train the next generation of clinicians and clinician scientists to study Alzheimer's and related dementias. And so every year we bring in a group of talented, promising younger investigators who either are already working in the ADRD space or would like to work in that space, and try to facilitate their maturation into independent scientists who are really making cutting-edge contributions to brain function, dysfunction, dementia, et cetera. And so today we will really be focusing on the trainees this year from the research education component. I will also mention that we have a couple other initiatives that are part of our center. We have a wellness initiative, and we also have the Lewy body dementia sort of a support group initiative for Lewy body dementia as well. On the upper top part, we have a Detroit advisory group, which is an important guidance group for us in the city, in the area of Detroit. Our center has a strong emphasis on understanding more about what happens in people who have cognitive impairment, who are underrepresented minorities, particularly African Americans. And so a great part of what we do as a center is working in the Detroit area, focusing particularly on the African American population that has been understudied in the dementia space for quite some time. So the Detroit Advisory Group is providing us help there to make sure that we are approaching the problems in the most culturally sensitive and appropriate way. Next slide. The admin core I run together with Bruno, and it would not happen without the assistance of Ari Baumick and Nancy Laracy, who divide up the really valuable chores of an administrative core. Next slide. The clinical core is really the engine of an ADRC. It is where we really collect the data on people who have cognitive impairment or who don't have cognitive impairment but serve as important controls for us. Really, our goals there are to really facilitate, enhance cutting-edge clinical research, and really, I think, as I mentioned already, enhancing the diversity among the population that we study as a field in aging and dementia research. Next slide. The data core is really the binder of a center. It's the core that really allows the data to be collected and available to people and uploading the data to the NAC. I'm really pleased with what our data core has done in terms of making the data accessible to all in a very fast and user-friendly format. We have a public request form that is now being used to request data from the center, and we encourage you to think about that. Next slide. One of the things I think the data core has done very well is create a virtual connection that allows investigators across our 3 universities to connect to the data in a non-laborious way. And this is just one example among many now, and a growing number. This is Patrick Pruitt from Wayne State, who is able to access the imaging data basically remotely. He doesn't have to, collect the data and bring it over to his site. And he happens to be at Wayne State, but the same applies to Michigan State investigators. And ultimately, we think will apply to other academic sites within the state of Michigan. Our focus, again, is the 3 Regents universities right now in the state. But as you know, University of Michigan has several other satellite universities or locations in Flint, Dearborn, for example, and investigators at those institutions are eligible for access to our data too. Next slide. The Neuropath Core provides valuable brain tissue to participants not only locally but nationally and internationally. The people who we evaluate as their disease progresses, if they have disease and they die, they very often generously donate their brain tissue for the advancement of research. And so the brains are collected. There's an autopsy of the brain. There's a neuropathological assessment. So we really know in fine detail the status of those brains, and the tissue, both fixed as slides as well as frozen tissue, is available as biospecimens to people who have questions they want to address. This is vitally important, and the center is regularly giving out tissue around the country, not simply to our 3 universities, to enhance research. Next slide.
I think 2-minute warning, just so you know.
Thank you.
Oop, if I can get it, there we go.
It's a big brain bank. We have somewhere close to 1,500 brains. There's a range of diagnoses. I think this is almost a little out of date. A lot of them are AD, but you'll see that there are other forms of dementia, such as FTD, Lewy body dementia. We have a very large collection of FTD and ALS brains as well, and some control brains. Next slide. The Outreach, Recruitment, Engagement Corps put together this symposium and is really vitally important for connecting across the entire state. Next slide. We have the initiatives, which I've already mentioned to you. Next slide. Now, the research education component is really, the goal is to recruit talented and diverse junior investigators into interdisciplinary dementia research and to enhance their research and career skills. This is a list. This is showing some of our investigators you're going to hear from today. Next slide. We have an online dementia curriculum, the Dementia for Scientists. This is something that you can access on YouTube channel, and they're quite good. They're only about 20 to 25 minutes long each. Next slide. As I mentioned, new cores under development. We're very pleased that Michigan State will play a vital and central role in the biomarker core, and the imaging core will not only do MRI and fMRI, but also PET ligand imaging to really enhance what we know about the brain involvement in people that we study. Next slide. So there are a lot of resources available to you. We do pilot grants. You'll hear from some investigators today who are doing research. If you have an interest in ADRD research, do not hesitate to contact me directly or any of the core leaders or even Aaron. Next slide. Okay, next year we're having our actual symposium from this year. This is our virtual symposium. Next year we're going to have the Beyond Amyloid Research Symposium in Ann Arbor. It'll be on June 10th. Save that date. Next slide. I think we have time for a question or two if there are any. I know that was a whirlwind overview of the center, and many of you know about the center. Some of you may not, and my goal was really to give you some sense of what we do and what we want to do for you.
Hank, this is Amara. Just a quick question about wondering what your success with getting individuals to donate brain is relative to the population you follow.
Yeah, I think it's a really big question. You don't start to evaluate someone and say, we would love to have you participate in research. By the way, we'd like your brain when you pass away. It's important for people to gain trust in what you do. So I think it's really a connection that we make over time. And in general, people who have participated with the center for more than a year or two understand that goal and usually say yes. But almost everyone that we have started to evaluate in the last couple of years says they are considering it. And the success rate, if you will, is quite high. I can't give you a percent right now, but it really depends also. And I would not anticipate someone on day one saying, yes, I'm definitely committed to giving you my brain 8 years from now. But we have had a pretty good success rate. And I will say, Amara, it's important. When we collect longitudinal data that includes neuropsychological data and imaging data, that data becomes super valuable when we actually know what the brain looked like in the end. And I think our research participants get that point, and they are so generous in their donation of their data, if you will, to advancement of the cause that they most often say yes, they are willing to donate their brain. Good question. Well, I would say to anyone else, you feel free to send me a question directly by email, or you can always call the center. I think we should move on, Erin.
Great.
What we're going to have now is a series of presentations from our current REC trainees. Now, I will say to the REC trainees, some of you have a lot of slides. You have 13 minutes, and Erin's going to give you a 2-minute warning, and you're going to be shocked to find that you're only halfway through. So I encourage you to zip through what you think are extraneous slides to get to the meat of your presentation. Our first presentation, I'm pleased, is Amara Ezeamama from Michigan State University. I'll turn it over to you, Amara.
Great. Good morning, and thank you for this opportunity. I'd just like to talk about work in progress on HIV and psychosocial stress in terms of neurocognitive disorder in a wide range of Ugandans from the work we've been doing in Kampala. Next slide, please. So just to remind, and it's okay, you're fine where you are. So yeah, so my goal for the REC fellowship or program for me was to really try to make this transition to authentically engaging in subjects of aging with HIV and understanding how some of the unique aspects of that for people who are HIV positive, but really getting into the issues of cognition impairments, which has really been the thrust of my research to date. And so my goal is to leverage that in basically writing persuasive proposals that are not pigeonholing me into an investigator of HIV. So today is one of the updates on one of several studies that we're doing, looking at HIV and stress-related differences in the global and domain-specific cognitive impairment among Ugandan adults. Next, please. I happen to be fortunate to be advised by really top-class investigators who are leaders in their field, and they really have been robust and contributed a lot to this work. Next, please. And can you skip to the next, and I'll just, thank you. So, HIV positive individuals are at special risk of neurocognitive disorder, at least compared to HIV negatives. And consistently, you find reports of deficits in cognition relative to community controls. But there are several questions that remain. And first, with the widespread use of antiretroviral therapy, especially in the developing world, you now have a phenomenon where the most severe forms of neurocognitive disorder, HIV-associated dementia, has become much rarer. but yet the mild forms of cognitive disorder have increased. So that's one aspect of the HIV transition that is unique, where people are living longer, mostly healthier, but more cognitively impaired. And yet, throughout the HIV epidemic, while psychosocial stress or some measure of psychosocial adversity is not unique to HIV, it is a phenomenon that is truly magnified in people who are living with HIV, often as a function of HIV-related stigma, and it is not something that is unique to populations in sub-Saharan Africa. Questions do remain, though, about whether the cognitive deficits that is often reported rise to the level of disorder. And we're particularly interested in understanding aspects of how the HIV-specific differences may be modified by psychosocial stress. So our hypothesis is that neurocognitive disorder would be higher in HIV positives, and higher psychosocial stress would correlate with neurocognitive disorder regardless of HIV status. Next slide, please. So this study is a cross-sectional analysis of data that we generate from 2 cohorts that we've been fortunate to follow, to start and follow in Uganda. One of them is completed. It's a pilot study that was pretty large, of 219 age, sex, and village matched HIV positive patients that are matched with HIV negative controls from their same communities. And then we also have an ongoing study now in follow-up phase of that includes adult caregivers of HIV positive, HIV exposed, and HIV unexposed, uninfected children. Together it's about 488 adults that are between 20 and 87 years old. Next, please. So we enroll individuals of known HIV status by design. So these are tested and known HIV positive status of the adult caregivers or the individuals who have been in care. For those who are HIV positive, they typically are not having recent disease. They've been connected to care, typically on average for somewhere around 9 to 10 years at least. So, and many of them for much longer. And we do define, so that part is standard, is clinically defined and confirmed. And then we have this measure of adversity, which I call stress, and that we basically define as the sum of adverse events out of 20 maximum, that using a standardized tool, the Recent Life Events Questionnaire. We ultimately analyze that in categories, basically looking at those who have the least events compared to those who have the most events. And there's 3 categories in between. Next slide, please. So the primary outcome for this study is cognitive impairment, and I look at this domain-specific but also global measures. So there are 7 domains that are defined based on performance-based neuropsychological tests that are as shown. Generally, impairments we consider as performance that is 1.5 standard deviations lower than an age-sex standardized performance for the in an HIV-negative adult. This is a population where we don't have population norms do not exist for this test. So what we've done is basically internally standardize the HIV-negative controls within age bands. And so an individual is impaired if they're 1.5 standard deviations on the lower side. So next slide, please. We then use each of those specific domain impairments to determine global measures. And so the first of these measures is the sum of cognitively impaired domains. So we basically class them as individuals who have 1, 2, 3 or more domains that are impaired. And then a second measure of global neurocognitive disorder is basically based on severity of cognitive impairment. We use a modified Frascati criterion where we basically combine proficiency in instrumental activities of daily living and age-sex standardized scores that represent deficits on 2 or more domains. Ultimately, we get 3 categories that we define as the outcome. We have individuals who are unimpaired, not cognitively impaired compared to the reference that form the reference for this study. And then we have asymptomatic neurocognitive impairment, and then minor or major neurocognitive disorder these categories are combined for to get to more power in the most severely impaired category. Next slide, please. So we conduct both descriptive and multivariable analysis, and I'll show you some data shortly, but try to describe the prevalence of disorder by cohort, but also stress measures, and also by cohort as well as by HIV status. And then we a lot of confounding variables that we take into account as well. We combine, we then implement multivariable analysis that are basically looking at HIV and stress-related differences or odds of impairment in the specific or global cognitive domains. And these measures, because they're either logistic, 3 levels or 2 levels, could be binary logistic regression or proportional odds type models. We adjust for some factors, and then we look for interaction that we think is present or warrants further investigation when the p-value for the HIV and stress interaction is at 0.1 or lower. Next slide, please. So here I'm showing results because I just told you that I combined 2 cohorts that are similar in the tests that we implement, but then they're different in some ways. So our study in the mild May cohort includes individuals that are on average, most of them in both cohorts are females. It was just harder to get men to say yes, but one of them in the Koala cohort is by design, where it's primary caregivers. But in both cohorts, you see that we had more success getting men to say yes, at least in mild May. The average age of individuals were quite different across the cohort. So individuals in mild May cohort were much older by design, because we only enrolled those who are 80 years and 50 years and above. So they're about 18 years older on average than those who are in the Koala cohort. And stress measures were interesting because in terms of acute stress, you don't see much difference. That this is acute stress defined as amount of stress experienced within 30 days. But in terms of recent life stress, we see that there seems to be higher levels of recent stress over the past 5-year period in Koala, which is a little bit much of a poorer area compared to mild May. So we think that it's probably socioeconomic status. But then when we look at lifetime adversity, you see what I think is a measure of lifetime experience, with much higher events in mild May cohort than in the Koala cohort. Educationally, there's more, the population in mild May is a little bit more educated. Next slide, please. Okay, so here we are basically looking at the percent that are impaired by cohort. I didn't have a good way of showing this on one slide for both cohorts. So this is for the mild May cohort. So if you look at motor speed impairment, for example, among HIV negatives is 15.8% compared to 21% in HIV positives. Not different. You see an average that for most outcomes, so going executive function, processing speed, simple attention, you see a trend towards greater burden of impairment in HIV positives compared to HIV negatives. And then no difference in terms of working memory, and the same a burden of higher level of fine motor impairment for positives relative to negatives. Then if you look at the average number of events, total domains that are impaired, you see on average about 2 domains impaired in HIV positives compared to 1 domain. And the severity of cognitive neurocognitive disorder is much higher in HIV positives in the Koala cohort. I'm sorry, the end of this slide is not showing very well, but basically you see, but if you look at the most severe category, you see that major minor neurocognitive disorder, 58% of those who are HIV positive are in this range compared to about 28% for those who are HIV negative. Next slide, please.
2-minute warning, Amara, just so you know.
Okay. Next slide, please. And you can skip this this is just similar data on the mild May cohort. We're just going to go straight into the multivariable analysis. So here I'm showing HIV-related differences, odds of cognitive impairment for the variables where, for the outcomes where there was no heterogeneity or no interaction with stress. So on average, HIV positive adults, the odds of cognitive impairment was twofold higher in the domains of motor speed, executive function, processing speed, and working memory. And these are adjusted for some confounders. Next slide, please. And then in terms of stress, we found a pattern of lower odds or protective odds, a protective association with impairment for those who are the lowest stress level compared to the highest stress level. I'm just showing two outcomes that illustrate this. So in terms of executive function, you see the odds of impairment is 0.3 times lower for those who are who have the least amount of stress compared to the highest amount of stress. It's not exactly dose dependent, but you get the trend that less stress is beneficial for cognitive function. And then not always statistically significant, but the trend was interesting. Next slide, please. We found interesting interactions between stress and HIV for some cognitive domains. So this is where the relationship of HIV status varied according to the level of stress that the individuals reported. So the bottom line story is that in the environment of high stress, the HIV negative status advantage we see in cognition is lost. So that, but when you look among individuals that report low levels of stress, you basically see that the HIV negatives have a cognition advantage in these respective domains. Next slide, please. This is another way of just confirming the same data that is basically showing that HIV negative individuals are much more sensitive to high levels of stress. So that when you look at those who have low levels of stress compared to those who have high levels of stress, and here I've just dichotomized, you see a protective association with that being significant in a few domains or global cognition as well. Next slide, please. So just to summarize, HIV positive compared to HIV negative had about 2 times the odds of impairment in more than one cognitive domains that we looked at, and I just listed those here. And then low psychosocial stress, on the other hand, was a protective risk factor for cognitive impairment, and this is especially true for executive function. We see these differences in relationship of these 2 factors to cognitive impairment for some domains, and with, next slide, please. And when you basically examine the nature of interactions, you notice a pattern of HIV negative adults being much more sensitive to the adverse impact of stress. And then when you basically put them in, look at those who are in high stress environments, that's protective association or that advantage of HIV negative status is lost. Next slide, please. And so this is an early analysis, and it's cross-sectional, so causal inference is not is limited in this context, but it's quite interesting nevertheless. And we do still have a lot of work to do in terms of standardization of cognitive test scores. I'm working specifically with Leah Rubin to think about ways of standardizing the test scores in our cohort to similar tests and performance tests that they've done in the Rakai cohort, which is a larger cohort also in Uganda. I didn't have the time to adjust for comorbid disease yet, so that is pending, and these results are confounded by that. And we're specifically interested, I'm very interested in looking at the potential for synergy between aging and HIV, and this analysis doesn't get to that, but it's a future direction where we want to examine for those who are older with HIV, what is the evidence that cognitive impairment or dysfunction is accelerated or accentuated compared to HIV unexposed, HIV negative younger individuals. Next slide, please. And so the conclusions are that people who are living with HIV that are older experience higher, at least adults with HIV experience higher rates of disorder compared to HIV negative peers. And stress is associated, lower stress is cognitively sparing, at least in some domains. And there was interactions that we're still exploring further. So multimodal interventions that mitigate the impact of stress in combination with potentially cognitive rehabilitation therapy might improve cognitive function regardless of HIV status. And in some cases, we're impressed that it looks like HIV negatives would also benefit a lot as well. I'd be happy to take any questions. Thank you.
Thank you very much, Amara. I really appreciate it. It's very exciting, and clearly there's more work to be done, as you mentioned, when it comes to comorbidities. I can't help but wonder, in this area of COVID-19, obviously the coronavirus that we're talking about is a completely different virus from the HIV virus, but the idea of stress and the relationship to viral infection or chronic disease, have you begun to think at all about how the COVID-19 infection or the way it affects our lives also could be a contributor to cognitive impairment? It's something as a center we need to be thinking about as we move forward, and that'll be true not only in the United States but in Africa. Any thoughts about that? I don't want to take you away from your own work on HIV, but you've been thinking about this issue, and I'm sure you have good thoughts in that regard too.
So we've been looking at some of the opportunities as well to see how can we move into that space, but definitely even from the aspect of psychosocial stress. So I've been in touch with with colleagues who are working in the field, and the study has figured out ways to move on. And participants who are living with HIV are very concerned. So even if they're not infected with COVID, the stress levels go up in that regard. However, they may be they're definitely at much higher risk of acquiring infection. And so we have not yet thought about how we can nest those testing for HIV, for COVID in this study, but it's something that we are eager to also explore. Because it ties in so nicely with some of the things we're thinking about, not just the biological effect of the virus, but also the stress-related dimensions of it as well.
Thank you. Other questions for Amara?
I have one. Hi, this is Shannon Moore from the University of Michigan. That was a very interesting talk. I really appreciated it. One thing I was wondering, I might have missed when you said this, but so you're lumping psychosocial stress or stresses sort of together. And I was wondering, do you have any plans to sort of break those up into different, like what kind of stresses are you talking about? And do you have any plans to break that up into different, so like the death of a family member versus low socioeconomic status versus, getting sick with another, a cold or coronavirus or whatever. I just was wondering if you could talk a little bit about the different types of stresses, and if you might look more fine-grained at those and how they interact with the cognitive impairments.
So Shannon, thank you for that question. So clearly one of the things, I'm looking at, we're using the recent life events questionnaire and probing for the occurrence of events. In this questionnaire, it's about 20 unique events over the past 5-year period. So we're looking at serious illness, death of a family member. We're looking at housing security or movement in the past 5 years of homes due to choice or not. And it goes on and on. So those are the kind of events. We do distinguish clearly from other legitimate stressors, like while it talks about job loss, we control for education and other, do you have an income or not? In some ways we may be over controlling for some dimensions of stress. But in our other work, where stress is also a big measure, we actually go ahead and we measure physiologic stress in the allostatic load framework. So, but it can definitely be analyzed a lot more finely. So your point is well taken.
Thank you, Amara.
Thank you.
I think for the sake of time, what I'm going to encourage people to do is directly contact Amara if you have questions or you wish to collaborate with her on projects in HIV or other areas. And I think we will move on. I appreciate that very interesting presentation. Our next presentation, I don't know, Aaron, if you can, is from Daugherty at Wayne State University. And I will just turn it directly over to Anna if she's ready to go.
Sounds good. Thank you for the invitation to speak today. I appreciate the opportunity to talk about my research program that uses MRI biomarkers to study vascular and metabolic risk factors for cognitive decline and aging, as well as Alzheimer's disease-related pathologies. Next slide, please. 1 in 3 adults has hypertension. Incidence correlates with age, and by age 65, over 60% of adults in the US have a diagnosis of high blood pressure. This substantially increases the risk for Alzheimer's disease and related dementia. Someone who is diagnosed with hypertension in their 30s or 40s is at twofold risk to develop Alzheimer's disease decades later. Because hypertension is a risk factor that presents in midlife, we have an opportunity to intervene and possibly slow the neurodegenerative process. There are many ways to control blood pressure through lifestyle factors, pharmaceuticals. The cumulative evidence suggests that successful blood pressure control does not completely mitigate the risk for Alzheimer's disease in later life. This risk disproportionately affects Black and Hispanic elders who are more likely to be diagnosed with hypertension and less likely to achieve blood pressure control as compared to white counterparts. And these racial and ethnic minorities are at increased risk for Alzheimer's disease and related dementias. So we know that hypertension is a meaningful risk factor for Alzheimer's disease that presents in midlife, and that current therapeutic strategies do not completely mitigate this risk. There's a gap in our understanding of how hypertension interacts with the neurodegenerative process. This is a simplification of that process. There are microstructural changes in the brain, including metabolic decline and oxidative stress, that contribute to declines in brain function and structural atrophy. And these together drive cumulative cognitive declines in aging, and even more so in diseases like Alzheimer's disease. Hypertension has been hypothesized to interact with this system to exacerbate the typical aging process and predispose people for a neurodegenerative disease. But individuals who have a diagnosis of hypertension and successfully control their blood pressure still show an accelerated rate of decline. This suggests that there must be another factor at play that propagates this neurodegenerative mechanism and interacts with hypertension. I hypothesize that one factor is iron accumulation. Non-heme iron is stored within cells to participate in normal metabolic processes. It's required for ATP generation, DNA replication. In the brain, it's required for neurotransmission and myelinating processes. In the process of aging, non-heme iron accumulates outside of binding complexes to produce free radicals, and in excess, this constitutes oxidative stress. Oxidative stress in turn impair mitochondrial function and accelerate metabolic declines, and these factors together interact with iron homeostasis to further accelerate iron accumulation. These bidirectional relations act like a self-propagating cascade, and much like an engine, it drives the cumulative and progressive declines in brain structure, function, and cognition across the lifespan. Histological and animal studies support that iron accumulation is an antecedent to the neurodegenerative process we believe is relevant to the effects of hypertension. We have an additional benefit that brain iron concentration can be measured in living brains using MRI methods. This is one example of an MRI technique called quantitative susceptibility mapping, also sometimes referred to as SWIM. On this type of image, darker intensity value corresponds to proportionally greater iron concentration. This is an example of an image taken from a middle-aged adult, and it features the basal ganglia prominently. These are regions that have high amounts of iron concentration across the entire lifespan and preferentially accumulate iron in the course of aging. I've used these MRI methods and similar ones to study the behavior of iron accumulation in typical aging, as well as its relation to neural and cognitive declines that may be sensitive to detect Alzheimer's disease-related pathologies. This is one example of a data set using a method called R2-star, and on the vertical y-axis, higher values indicate greater iron concentration. And as you can see, age strongly and positively correlates with greater iron concentration across the lifespan. In a series of longitudinal studies, I've shown that these MRI methods are sensitive to detect iron accumulation in aging brains over a few years. That brain iron accumulation precedes and predicts structural atrophy up to 7 years. Iron accumulation also predicts cognitive declines that are typical in aging and are sensitive indicators of Alzheimer's disease pathologies. For example, accumulation in the caudate nucleus specifically predicts declines in working memory and executive functions. And accumulation in the hippocampus specifically predicts declines in declarative memory functions. Pairing these methods with task-based fMRI, I've shown that greater iron concentration interferes with functional activation. And even in middle-aged adults, greater iron content diminishes functional activation and partially accounts for deficits on the task. So it appears that the MRI methods are tracking with the known histological action of iron accumulation, and that these MRI methods may be useful biomarkers for studying this neurodegenerative process. More recently, I've begun to evaluate its relation to other MRI biomarkers that may be more specific and sensitive to Alzheimer's disease-related pathologies, increasingly earlier in the lifespan. This is an example of a cross-sectional data set of middle-aged and older adults. They are in preclinical stages of cognitive decline, and they participated in a PET imaging sequence that allowed us to quantify beta amyloid burden throughout the brain using the index SUVR. And I'm showing you 3 panels moving from left to right of low beta amyloid, moderate, and high levels of beta amyloid based off of clinical suggested thresholds. The x-axis is R2 star, and higher values indicate greater iron concentration. The vertical y-axis is the relation between intrarenal cortex volume and age. Now, Alzheimer's disease-related pathologies begin in the medial temporal lobe, and the intrarenal cortex is one of the first regions that is sensitive to decline in preclinical stages. So a negative relation with age on the y-axis would indicate an accelerated neurodegenerative process and possibly signal Alzheimer's disease-related pathology. Moving from left to right across the panels, you can see that that slope is increasingly negative. And on the far right, among individuals who have the highest beta amyloid burden and high iron concentration, we find evidence of the smallest intrarenal cortex volumes with age. That relation is diminished with lower levels of iron and diminished at lower levels of beta amyloid. So taken together, in individuals who are in preclinical cognitive decline, it is the presence of both iron and beta amyloid burden that identify individuals who are likely at the highest risk for developing Alzheimer's disease-related pathologies in the upcoming years. This, of course, is a cross-sectional data set, and so I am very cautious in interpreting any causal relations. And as we follow these individuals longitudinally, it will become increasingly important to understand the temporal precedence of these MRI biomarkers. To summarize where we've been in this talk so far, I've shown you that these MRI methods appear to be sensitive to age-related brain iron accumulation, and I've described a series of longitudinal studies that suggest that brain iron accumulation precedes and predicts structural and cognitive declines that are typical in aging and would be relevant to Alzheimer's disease-related pathologies. The next question is if hypertension interacts with this system. And this is where my current research program is stepping in and designing a new longitudinal prospective study to follow individuals who have no history of hypertension, as well as individuals who have chronic hypertension, to track this disease process. Here are some preliminary data from that study. These are all cross-sectional and are only middle-aged and older adults who have no history of hypertension. And as you can see, even subclinical elevation in pulse pressure correlates with iron accumulation in the caudate nucleus and hippocampus. So what appears to be occurring is that iron accumulation indeed precedes and predicts these structural and functional declines, and based off a histological and animal study, we can be confident that it is an antecedent to the microstructural changes we hypothesize. Iron homeostasis is dependent on vascular and endothelial function, and even minor disruptions will acutely increase iron concentration in the brain. So it's plausible that chronic hypertension may modify iron homeostasis and trigger a cascade of events that initiates an increase in iron accumulation and then subsequently the propagation of oxidative stress and metabolic decline. Moreover, chronic hypertension in midlife may act as a risk factor that initiates this cascade and this cascade might continue even if blood pressure control is later achieved. These are the questions that my current research is attempting to address, not only in the prospective study that I described with preliminary data here today, but also with the existing sample in the Michigan Alzheimer's Disease Research Center. And these include individuals who have mild cognitive impairment as well as Alzheimer's disease. We've followed them longitudinally with serial MRI, and in those existing data, I'm attempting to ask the question if hypertension as well as other cardiovascular risk factors may exacerbate the rate of brain iron accumulation in that sample, and the extent to which that might mediate hypertension-related risks for cognitive decline, as well as diagnosis of MCI and Alzheimer's disease. Because brain iron accumulation appears to be a promising biomarker that we can measure by non-invasive MRI, and it's sensitive to detect changes in preclinical stages as early as middle age, pairing these methods with also a study of chronic hypertension may help us to understand the seeming paradox of interventions that seem to not fully mitigate risk for Alzheimer's disease, but also sources and pathways of risk that may differentially affect racial and ethnic minorities, and the tailoring of intervention strategies to try to help those individuals stave off further decline. I appreciate your time and attention today. That's the end of my formal talk, but I'm happy to take questions.
Thank you very much, Anna. We really are pleased also to see that you are leveraging the data that are collected from the ADRC longitudinal cohort in your pilot study that will be funded this coming year. So we're very pleased about that. We have time for questions. It's a very complex interplay of events here that's still being explored. And I'll open up to the floor. Questions for Anna?
Yeah, this is Charlie Fehl at Wayne State. Hey, Anna. I love your scheme for connecting these two really big phenomena of oxidative stress and metabolic decline, and then trying to figure out specific drivers of disease. Now that the Michigan ARDC has the PET imaging core up, have you thought about coupling like PET studies for fluorodeoxyglucose to take a look at that metabolic decline bit and then build it into your overall working model?
Yes, absolutely. Of course, PET imaging is kind of a rarity in these observational studies. And so any data, even in smaller data sets or smaller sample sizes, can be particularly informative. In my prospective longitudinal studies, I'm also using other methods like functional magnetic resonance spectroscopy in order to understand ATP generation even at relatively low resolution in the brain to try to co-localize these effects. For example, brain iron concentration varies across regions, presumably because of differences in metabolic demand, but also differences in neurotransmitter systems and myelinating requirements. And so based on that, we would expect there to be whole brain systemic issues, which is how hypertension is likely interacting with this process, but also regional vulnerabilities that would signal specific types of cognitive decline patterns. For example, the hippocampus has relatively low iron concentration, but it's particularly vulnerable to these sources of risk and especially vulnerable to metabolic disruption. And so although iron concentration in absolute terms is lower than compared to other brain regions, that region may be particularly sensitive to even minor fluctuations. So these are really great questions. As you can tell, this is a kind of a burgeoning field. The MRI methods that allow us to measure iron are relatively new. And so now it's about staging and setting up the set of studies that will really help us to inform and infer about these biological mechanisms.
Other questions for Anna? Well, let me just add that, as you said, PET is a rarity. It's something we are trying to patiently obtain on our research participants, both the amyloid and tau imaging. Charlie asked about, fluorodeoxyglucose PET imaging. Less of that being done, but we have a significant collection of PET and MRI from individuals who were evaluated a number of years ago in the center, in which these of metabolism could be explored. Other questions for Anna?
So, Anna, I had a
Jeff
question.
Murphy.
This is Jeff at University of Michigan. Where is the iron accumulating? So I know there's evidence that suggests it accumulates in plaques, but I also know that other things like manganese, which is used to enhance MRI, enters the neurons through calcium channels. Where do you think the iron accumulates?
This is a fantastic question, and we can turn to both histological and postmortem study and also animal models of diseases to try to address it. The MRI methods that I use, of course, can't get to this level of specificity, so we have to use these translational methods. Based on those series of studies, we know that there are two different functional forms of iron. There is heme iron in circulating blood. This constitutes the core of hemoglobin. And today I was talking about non-heme iron, that is iron that is not of blood but stored in cells. So non-heme iron is stored in most cell types for basic metabolic functions that I described, and when it is in a neutral or metabolic neutral state, it's stored within ferritin molecules predominantly. These MRI methods are mostly detecting concentrations that are bound to ferritin, and as we know, higher elevations of those bound concentrations correspond to shifts in metabolism as it plays out as I described. With that said, though, in age-related accumulation, it appears to be mostly occurring within astrocytes. And this is entirely consistent with our understanding of iron participating in an immune response and a privileged relation with inflammation and microglial activation. And I didn't have that piece of the puzzle illustrated in today's talk, but there's an entire line of my work that's trying to understand the relation between low-grade chronic inflammation that occurs in aging, as well as genetic predisposition for these disorders. And there is an overwhelming amount of evidence of a relation between inflammation and pro-inflammatory cytokines and iron homeostasis in cell modeling and animal models and so on. And now we're just getting the preliminary evidence in living adult brains. So the age-related effects are mostly astrocytes, although the relationship between beta amyloid deposition and plaques is very intriguing. There's a bidirectional relation there, and there is some now preliminary evidence that it may interact with tauopathy, although it's not very clear if that is a causal relation or merely just a confluence of changes that causes these different markers to co-localize.
Thanks.
Thank you, Anna. I think for the sake of time, we best move on, but I encourage all of you to contact Anna directly with further questions and potential collaborations. We should move on to our next presentation, and that will be Annalise Rahman-Filipiak. Annalise.
Thanks so much, Hank, and thanks everyone for being here. So today I'm going to be talking about some work I've been doing over the course of the past year, focusing on risk disclosure. Next slide, please, Erin. So as clinicians and clinical researchers, we tend to encounter patients and their families most often when they're already symptomatic. So in terms of the Alzheimer's disease cascade, they already have some subjective complaints and perhaps functional difficulties that lead to us disclosing information about most often their diagnosis of either MCI or dementia Alzheimer's type. However, we know from the ATN or the ATN-B model that certainly the Alzheimer's disease cascade happens much earlier, and we certainly have the technologies now to be able to identify potential markers that might confer later risk for developing dementia Alzheimer's type. However, unlike things like ApoE, which are being used in clinical settings and revealed and shared with participants in research in clinical settings, and certainly are available through direct-to-consumer methods like 23andMe, we really don't share biomarker profiles, things like amyloid and tau, that often in clinical settings. And most often this information is collected in research studies. However, we share it very rarely with our participants and their families. And that's for a multitude of very good reasons, not least of which is that we don't really know whether participants and their families are interested in knowing this information, and we're not really sure how they're going to use it, particularly in the context of a lack of validated treatments to slow or stop the decline of Alzheimer's disease. I think we have major concerns about sharing that information in a way that's going to be useful to our patients and their families. Also, there really aren't any kind of validated and empirically supported feedback protocols to share this information. Next slide, please, Erin. So as someone that is very interested in health disparities, particularly in dementia diagnosis and treatment, I'm also very aware that there are going to be contextual barriers that impede certain populations, particularly racial ethnic minorities, from using this information as adaptively as others. So for instance, we know that things like financial security, insurance coverage, cultural competence of providers, all of those factors may impede someone from really being able to understand and use this information as well as others. So I also have questions about whether risk disclosure needs and preferences differ in Black versus white older adults, and how these can be explained by known social determinants of health. Next slide, please. Of course, we're also interested in outcomes. So if we consider risk communication as an intervention, we want to know the safety and efficacy of that intervention. Does it increase mood problems or anxiety or even suicidality? And when we're talking about a population of individuals who tend to have cognitive difficulties, we of course are going to wonder whether or not they can understand this information and remember it over time in order to use it. Next slide. So all of these ideas were kind of percolating in 2018 when I wrote this R03, which was funded last year. And so I'm going to be talking today mostly about the first stage of this study, which is the needs assessment. And our objective is to evaluate the needs of Black and white participants and caregivers in regard to disclosure of their risk for DAT or dementia Alzheimer's type based on a variety or different levels of risk factors. The first would be clinical information, so the standard information that we collect in a clinical visit, things like family history, symptoms, etc. Second would be risk enhanced by information from structural neuroimaging. Next, we would talk about risk based on genetic status, and particularly ApoE. And lastly, and this is a particular question of mine, is biomarker-based risk. Given that this information is much more complex and perhaps less familiar to our research participants and patients, would they still want to know? And not only are we interested in whether people want to know, but also why they would want to know or not, how they would want that information shared with them, and again, how those social determinants play a role. And our hope with this mixed methods needs assessment is to kind of compile this information into a feedback protocol that is semi-structured and flexible to allow us to take more of a patient-centered and culturally sensitive approach to sharing this information with patients and their families from diverse backgrounds. Next slide, please. So this just presents kind of a timeline of the project. And so right now we're still in the relatively early stages of the needs assessment, but our hope is to collect 50 participant caregiver dyads. That the participant identifies a caregiver, and that's either someone that's currently serving as a caregiver or would be the loved one to serve as a caregiver were they to have some kind of cognitive or physical difficulty in the future. These are individuals that are all drawn from 3 different parent studies. So UMMAP, DAPA, which is another MADRC study, and the Stimulation to Improve Memory study run by Dr. Hampstead. And we chose these 3 studies specifically because first we know that all of the individuals are going to be well characterized and diagnosed using standardized clinical criteria. But also we know that all of the participants drawn from those studies, we checked first to make sure that they had already undergone all of the procedures that were necessary to collect all of the different biomarker or risk indicator information. So we hoped that by doing that, these individuals would, in talking about their decisions or their needs and preferences around risk disclosure, those would be less kind of hypothetical thoughts. All of the individuals are diagnosed as either cognitively normal or with amnestic mild cognitive impairment, and we really are striving to get an equal sample of African American and white older adults. They complete a single visit. So far that's been face-to-face, though with COVID we have just switched to a virtual format. And during that visit they complete some questionnaires that are sort of standardized, but they're also completing a semi-structured interview that was developed through concept mapping for the purpose of this study to really get at not only their needs and preferences, but also their reasons, health beliefs, those kinds of things when it comes to risk disclosure. Next slide, please. So I'm going to be sharing just a very brief snapshot of some of the data that we've collected so far through March. And we can go to the next slide. So this is from 20 of our participant caregiver dyads. And I just showed the demographics here to essentially say that we do at this point have relatively equal numbers of healthy older adults and individuals with amnestic mild cognitive impairment. And I should note that all the caregivers are cognitively intact. Next slide. So what I'm going to be showing, again, this is primarily descriptive information, but some of the first questions that we ask individuals are about their interest in receiving these 4 different levels of kind of risk information. And we ask this question in 2 different ways. First, we ask them about their interest on a 5-point Likert scale, ranging from no interest all the way to strong interest, which is the blue bars that you see here. Then we also ask them to make a dichotomous sort of forced decision. So today, if I had a packet of information with me, which has your risk based on standard clinical information, would you want to know it? Yes or no? And as you can see here, there was a pretty overwhelming response. So for both participants and co-participants, most endorsed a strong interest, and all of them endorsed that they would want to know that information right away or today. Next slide. This was also true, just go one more for me, Erin. So this was also true for structural neuroimaging. Sorry, go back a couple. I think we skipped a couple. But basically, you're giving away the great punchline here, which is just that, that's okay. But looking throughout the different biomarkers, we really saw a pretty strong interest regardless of what level of risk information we were providing. So while people had, particularly co-participants were maybe a bit less interested in receiving information based on structural neuroimaging, still 100% of participants and 95% of co-participants suggested they would want this information today. And moving to the next slide, we see that basically the same is true for genetic information. So co-participants a bit more hesitant, but still 90% of them would want that information and 100% of participants. And finally, when we look at interest based on biomarkers in the next slide, so interest and risk based on biomarkers, we see almost exactly the same story. And so, at the bottom, it's probably hard to see on some of your screens, but when we talk about group differences in interest, you can see there's very little variability here, and in fact, no differences based on race or diagnosis. So we can move to the next slide. So the other thing that we wanted to ask was about reasons. So we did this in a couple of different ways. We listed all the possible reasons that we could gather from the literature and from other specialists in the area, why people might want to know their risk for dementia Alzheimer's type when they were asymptomatic or had early symptoms. And we had them endorse as many as applied to them. And then we also had them endorse their main reason, which is what I'm depicting here in the interest of time. And we see that for participants, the majority of them are, there's a range of different reasons listed, but a lot of folks are interested in gathering this information to engage in treatments or clinical trial research studies, to modify health behaviors, and to inform long-term care plans. If we move to the next slide, which depicts the main reason listed by co-participants, we see that again, there's an interest in enrolling their loved ones or helping their loved ones enroll in clinical trial research studies, but also we see that a decent proportion of them are very interested in knowing about risk from kind of an adaptive or compensatory standpoint to inform long-term care plans. So in the next slide, we're going to see all the possible barriers that we listed. So again, this is from kind of our specialist panel as well as the literature. These are all the possible reasons why we thought people might be concerned. And certainly I think, again, as clinicians and clinical researchers, these are perhaps some of the concerns that we have in sharing this information. But I should note that we also had an open-ended sort of, is there another reason why you might be concerned about knowing one's risk? And certainly we also included a no reason or barrier at all, I would always want to know this information option. So the next slide is perhaps one of the most powerful findings, even in this just very kind of gross snapshot. So when we asked our participants to tell us about what barriers, all that apply, what barriers might inhibit them from wanting to know their risk for dementia Alzheimer's type, we see that only a few of them are endorsing concerns about the lack of available treatments or increased anxiety or worry if you did find out that you were at risk. Whereas 90% of them suggested that they would always want to know, that there was no barrier big enough to impede them from wanting to know this personal health information. Moving to co-participants in the next slide, We see that while they do endorse a range of other barriers and concerns, including increased anxiety or worry and lack of available treatments, 70% of them still suggested that they would always want to know this information and that there was no barrier that was going to stand in their way of that. So we can move to the next slide, which is conclusions. So, of course, we recognize that there's going to be a bit of a selection bias here in terms of who's participating in this study, and this is a very small sample. However, it aligns pretty well with the larger literature suggesting that participants and their loved ones report overwhelming interest in knowing their risk. And for participants, this seems to be because they want to be proactive about engaging in modifiable adaptive health behavior change, treatments or research studies. And for co-participants, it seems to be about planning, at least to some extent, wanting to compensate or adapt and plan as a family for the future. It's also important to note that while we as clinicians and clinical researchers have real concerns about sharing this information, the barriers don't seem to be as important to patients and their caregivers. And I think that really highlights the ethical dilemma that we face when it comes to risk communication between wanting to do no harm and protect our participants and patients, but also not wanting to be overly paternalistic in restricting access to health information that could create adaptive change. So moving on to my final slide here, in terms of future directions, we do have 30 more dyads to run, hopefully fingers crossed, in this virtual format. We hopefully at that point will be powered to look at the more nuanced aspects of social determinants. So rather than making this a simple black versus white issue, looking at factors like income, economic stability, insurance coverage, comfort with your providers, health beliefs. We'll then move on to the exciting part of the study, which is the risk disclosure piloting towards the end of the year. And then I recently submitted a career development award, which will hopefully extend the piloting process, but also look at long-term changes in adaptive behaviors over 2 years following risk disclosure. So I just want to say thank you. My next slide, thank you so much to my wonderful REC mentoring committee, Ben Hampstead, Scott Roberts, and Laura Zahadny, as well as Shima Sadagiani, who is my research assistant, who is my undergraduate volunteer, looking for a job next year if anyone's looking, and also, of course, funding sources, the parent recruitment studies, and our participants.
Thank you very much, Annalise. This is really exciting and important work. We'll open it up for questions. As you know, I always have questions, but I'd rather hear from other people with their questions.
Annalise, this is Darren Zoronic from University of Michigan. I'm sorry if I missed this, but were they told about the downsides of getting this information before you told them that? And what were they told? Were they told about, they might not be able to get life insurance, risk of suicide? Sure. What were they told?
Sure, that's a great question. So we went back and forth about whether we should provide significant education about the risks and benefits at this stage. And we decided that while for, of course, the actual protocol piloting, there will be a stage before they even consent to receive their risk information that's just purely psychoeducation that includes a review of risk and benefits. For this portion, we wanted to get a sense for what people knew, and many of the questions that I didn't go into today in the shared interview or semi-structured interview tap into what they know about the risks and benefits. And so part of this is just trying to get an understanding of what the large public knows about risk communication, about different technology. And that would really help shape our education portion for the protocol.
Thanks.
Sure.
Other questions for Annalise?
This is Judy Heidebrink. I have a question about what the participants were led to believe about the sort of quantitative aspect of risk disclosure. Were they implied that they might be given a specific risk number, or just sort of a general feedback about more at risk or less at risk based on any one particular piece of risk or the combination?
Yeah, that's a great question. So certainly we talked about this as qualitative risk. So we know that at this stage, there really aren't established risk curves, particularly for amyloid and tau, those dynamic biomarkers, where we can give them a specific number. And so we were talking more about being at increased risk or not at increased risk. So very gross and general information, which I think, again, that's part of the concern in sharing this information is not being able to be specific and knowing the complexity of particularly these dynamic protein biomarkers, how we share this information, knowing that it may change in 5 years for some folks.
I think for the sake of time, we probably should move on, but I did want to say that I think it's a new era in research and that providing information back to our research participants in a participatory way is critically important, and I'm glad that Annalise is leading the push toward that. So thank you, Annalise. Our next speaker is Charlie Fehl from Wayne State University. You've already seen him because he asked a question earlier. I'll turn it over to you, Charlie.
Okay, thanks so much, Hank. So I'm in my second year in the chemistry department at Wayne State University, and so this talk is going to certainly fall more on the chemical model system, where we're developing chemical tools to study a metabolic aspect of neurodegeneration or neuroprotection. And I'm hoping that our ultimate goals are to move beyond the chemical bit and go toward kind of some lifestyle outcomes of diet, intermittent fasting, and then exercise as ways to ameliorate some of these symptoms. So if we go to the next slide, my goal with the REC program is really to connect with disease biologists so that we can take our chemical tools into that next phase. And so I'd like to introduce the main system that we study, which is a glucose-related post-translational modification on proteins. Similar to Anna Daugherty's talk, in situations of metabolic and cellular stress, we get alterations in signaling patterns. And here I'm going to talk about a glucose-derived sugar called GLICNAC, which gets added to hundreds of different proteins. As glucose enters the cell, a portion, about 3% of it, gets converted into this UDP GLICNAC substrate for a GLICNAC transferase. You can see that enzyme on the green squiggle kind of in the middle. There's a single enzyme in humans that transfers O-GLICNAC onto hundreds of different protein, serine, and threonine residues to kind of change the way that they look and respond within the cell. And then there's a single enzyme in humans that takes that off called O-GLICNAC ACE. We can see on the right that if we dose a cell with a low or a high amount of this GLICNAC compound, we can stain hundreds of different proteins. And so these are involved in signaling processes, enzymatic activity, interactions, and what I'm mainly going to talk about today is protein accumulation. And so this O-GLICNAC levels and modification inside a cell allows cells themselves to sense their nutrient and stress environments. And they'll respond to oxidative stress, metabolic stress, as well as the presence of A-beta plaques outside the cell. What we are interested in is studying the hypoglucose metabolic effects in neurodegeneration like Alzheimer's disease and spinal and bulbar muscular atrophy, where a long-term goal will be to develop strategies to ameliorate these. So if we go to the next slide, we'll see a little bit of what that looks like. I'm showing you some data from other reports showing that the hippocampi, which are very much central to Alzheimer's disease progression, are addicted to the sugar GLICNAC. And if you knock out the transferase that adds GLICNAC to different proteins in mouse hippocampus, on the left, we'll see that even in developing mice, you essentially get instant neurodegeneration when you lose this enzyme in the hippocampus. Now on the right, on the flip side, if we add a chemical inhibitor that inhibits the enzyme that removes this modification on proteins and boosts the levels in hippocampi, we reduce the levels of pathological tau species. And there was a study in 2014 that showed that these also tracked in mouse models of cognitive impairment to great improvements associated with blocking tau aggregation. So the loss of this O-GLICNAC transferase can cause neurodegenerative phenotypes. If you boost the levels by inhibiting the enzyme that removes this, O-GLICNAC ACE, OGA, that can be neuroprotective. Some possible mechanisms there are directly inhibiting protein aggregation of these toxic proteins, increasing protein chaperone activity and protective signaling, but I think it's very multifaceted based on the fact that hundreds of proteins are modified by this process. This has culminated in the FDA approval for progressive supranuclear palsy, which is a tauopathic disease of a compound similar to thiamine G, which inhibits O-GLICNAC ACE and kind of prevents this as a neuroprotective fashion. So if we go to the next slide, we'll see that the challenge of using these compounds is that they're really all or nothing, because there's only one enzyme that adds and one enzyme that removes this, this modification. If we block it at a certain level, this is affecting hundreds of cellular processes. I'm showing some data from PC12 cells, which is our model of neurodegeneration and a neuron that we can culture in our lab, that we can very readily boost or inhibit O-GlcNAc levels in general using a couple of tool compounds, but there's a need to control specific pathways here. If you go to the next slide, we have a working model of some of these neuroprotective effects, where if you have a normal or high amount of cellular glucose utilization, we'll have the ability for the cells to put this GlcNAc modification on its intracellular proteins. But if you lose this, that can be neurodegenerative. Conversely, if you boost this, that can block the formation of toxic misfolded aggregate proteins. And so we're interested in looking at models of Alzheimer's disease and spinal and bulbar muscular atrophy, because both of these are characterized by the accumulation of intracellular misfolded protein aggregates. And these can block the glucose metabolism and thus the GlcNAc levels of these disease cell types that leads to cell death and ultimately neurodegeneration. So our key questions are, what are the key intracellular mediators of this glucose hypometabolism and neurodegeneration? And are they conserved between different types of neurodegenerative disease? So if you go to the next slide, we have an overview of what my group does. We implicate O-GlcNAc effects in disease biology. And then we're working on two different tools that will allow us to actually unpick what the specific drivers of these effects are. One is a photochemical tool where we can control the levels of intracellular GlcNAc. The other is a biosensor that tells us which proteins are O-GlcNAc-lated in a cell. So if you go to the next slide, we can talk a little bit about our disease models. Like I said, we use PC12 cells as a readily culturable neuron. It's a poor model of neurons, but it's kind of the standard in the chemical biology literature to study tauopathies and other neurodegenerative diseases before moving on to more advanced models. And so we have a tau mutation that is aggregation prone that we use for an Alzheimer's disease model. And then we, just before COVID-19 struck, we initiated a collaboration with Andrew Lieberman's group at the University of Michigan to study some of his PC12 models of SBMA, which is a polyglutamine expansion repeat disorder. We can see that in PC12 cells that O-GlcNAc is highly implicated because multiple pan-O-GlcNAc antibodies will stain these things. We're interested in asking what are some of the intracellular mediators here. And those, some of our early data is done by one of my graduate students, Zahid, shown in the lower right. So if you go to the next slide, one of the ways that we study these proteins is a system called metabolic labeling, where we essentially trick a cell into using a chemically tagged version of GlcNAc to put onto its intracellular proteins. This can label hundreds of intracellular proteins because the endogenous transferase, the O-GlcNAc transferase, is able to put our modified sugar onto a protein, and then we can attach a dye to this for visualization on the gel. The problem with doing this in a non-controlled fashion is by adding a large amount of this extra sugar, you bias all of the intracellular pathways toward hyperglycanylation, which is actually not really what's going on in Alzheimer's disease when we have a reduction of this. It can also be toxic when used at high levels. So if you go to the next slide, our solution to this problem is to create a chemically controlled variant of this, where we attach something called a photocage onto these molecules. Now the photocage keeps it from being active within cells until we treat it with a specific wavelength of light to release this. You can see on the left side of our plot, if we have an uncontrolled GlcNAc molecule, which has that red data, compound 10, that shows a high amount of labeling. That's that third lane on the gel from the left. Conversely, if we cage our molecule with a violet light, a purple light selective photo cage, the final 3 lanes on the right-hand side show a time-dependent release of these compounds and corresponding GLICNAC levels in cells. We've also shown that these are non-toxic compared to the parent non-caged compound. So we think they'll be nice tools for allowing us to study the time-dependent processes of neuroprotection in these neurodegenerative models. So our next step is to take these controllable GLICNAC tools and then really study the dynamics of protein aggregation and see if we can use these to work out the sequence of protective events in our PC12 model. If you go to the next slide, we also have a biosensor that we've created to identify what these proteins look like in cells. Our collaborator, Andy Lieberman's lab has reported some transcriptomic data that shows a number of metabolically important GLICNAC glycoproteins that are affected in SBMA, polyglutamine expansion disease. And so we'd like to identify what these changes are between the diseased and the healthy cell states. So we've put together an GLICNAC binding domain onto an enzymatic proximity labeling domain to create these biosensors we call glyco-ID for identifying glycosylated proteins. The binding domain will bind GLICNAC-related glycoproteins, and the proximity labeling, we use a system called BioID, which attaches a chemical label that we're able to fish out these proteins for identification using proteomics. So we've created these biosensors. We transfect our cell lines, either HeLa cells as a control or our PC12 cells as our neuron mimics with these biosensors. And ultimately, we'd like to compare our neurodegenerative models to look at what glycoproteins are involved. If you go to the next slide, you'll see that we've put these in cells. If we do a transient transfection with either just the biotinylating proximity labeling enzyme itself, which is shown in red, this expresses very well and it labels, as expected, essentially everything that it bounces around inside the cell and comes into contact with. But when we use our glyco-ID construct that are targeted to GLICNAC proteins, which I show under the blue bars in our gels, we see that we've indeed created our larger fusion protein on our expression data. And on the right-hand plot for our activity data, we see a very different pattern of labeling with our targeted versus our untargeted constructs. We're interested to next use proteomics to confirm that we are indeed labeling GLICNAC glycoproteins for specificity. You'll go to the next slide. One problem with
warning, Charlie, just so you know.
Perfect. We've also made it stable versions of this that allow us to look at the time course of these events. So if you go to the next slide, I can just quickly summarize what our group does. We study a neuroprotective role of GLICNAC levels in protein misfolding-driven neurodegeneration, and we've developed tools to allow controlled levels of GLICNAC in cells, as well as biosensors to determine which proteins could be the drivers of this neuroprotection. And so our goals with the Madrc Rec Program are, we've already initiated a collaboration with Andy Lieberman, and he has some of our compounds. Unfortunately, COVID hit before we could really test them. And I am seeking more guidance on Alzheimer's disease models beyond our PC12 tau mutant. We think we can find some more advanced models of this. So if you go to the final slide, I'd just like to acknowledge my lab. The graduate students did this work, as well as our collaborators, my rec mentors, Andy Lieberman and Irving Vega. So thank you very much for your attention.
Thank you, Charlie. Really appreciate that fascinating treatise on sugars and disease. Events, sort of glycation end products that have been described in age-related disease. Just quickly, just for the edification of our audience, what's the relationship between the GLICNAC events that you're talking about and those RAGE products that people have talked about for years?
Oh, that's a fantastic question, because both are stoichiometrically related to the levels of glucose in a system. And so people with kind of hyperglucose metabolism or diabetes or kind of larger body mass indexes can have correlations with these non-enzymatic post-translational modifications called these RAGE compounds where sugar derivatives actually get broken down and attached to proteins. These can be immunogenic and create, it looks like a foreign species to the body, and it'll accentuate inflammation. Now, GLICNAC is, by contrast, an enzymatic addition where it's a very controlled fashion, and the brain, because it uses so much of the body's total glucose, has very high levels of GLICNAC in neurons, as well as the associated enzymes that add and remove them. And so, as I said, the hippocampus in particular is especially sensitive to GLICNAC levels in a survival mechanism, if you will, to keep hippocampal cells operating physiologically, whereas the end product of glycosylation is more of a toxic buildup of glucose-derived modifications that stimulate an immune response.
Thank you, appreciate that. And there I go, asking questions again. Any other questions for Charlie?
I had a question. This is Sean Ferris from University of Michigan, ADRC Neuropathology. So are tau and A-beta modified by O-GLICNAC, and how like heavily are they?
Yeah, so tau is certainly modified, and these are intracellular processes, and so only tau tangles are going to be modified by O-GLICNAC. The A-beta, I don't believe they have good data that shows that gets modified and then exported before aggregation. But the main drivers of therapeutic effects of increasing O-GLICNAC levels are all based around the tauopathic diseases, because it directly actually inhibits the phosphorylation, the hyperphosphorylation of tau, which contributes to its aggregation and then the toxicity. And so if you remember my working model, we think that these aggregation-prone species that are not modified can be protected against aggregation by this O-GLICNAC modification, which acts as kind of a Teflon to keep them from sticking to each other. And there's even been an antibody developed against GLICNAC-related tau that is used pretty readily in the literature as a biomarker. So we definitely know that tau.
Charlie would add that Hannah Trzecikiewicz will be joining our group as a new postdoc on June 1st. She works on post-translational modifications in tau, and I would encourage you and Hannah to be speaking about ways that maybe perhaps we could be looking at tau and sugar modifications along the lines that you're looking at.
That's fantastic. I'm also making a note to reach out to Anna Daugherty, because a lot of the pathologies that we're studying seem to interact very nicely just between glucose levels and iron levels.
Super. Excellent. Well, I think for the sake of time, we should move on again. I encourage everyone to contact the REC trainees who are in the midst of their studies. Our next speaker is Ilse Medina Meza from Michigan State. Ilse.
Hello, can you hear me?
Yes.
Okay. Thank you so much for the introduction and the opportunity to participate in this great and magnificent group. I will present the dysregulation of cholesterol metabolism and its implication in brain aging. My laboratory is a pretty young lab. We have almost 2.5 years in Michigan State University. We are an interdisciplinary group with chemical engineering and biophysics studying the lipids, its derivatives, and its impacts in the life process. Next slide, please. I will briefly outline this talk. I will start talking about the lipid metabolism and its impact on brain health. Then I will discuss the cholesterol in brain and cholesterol homeostasis. I will explain also what are the oxysterol, if there are metabolites or biomarkers. And finally, I will be talking about the P450 cytochrome family. Next, please. So as we know, there is a variety of lipids that are involved in several complex and specific physiological functions, such as energy storage, cellular transportation, and formation of the cell membrane. Lipids are classified into the 5 major groups, starting with esterols, glycerophospholipids, glycerolipids, sphingolipids, and fatty acids. Lipids has been well recognized as the beneficial molecules for the brain health and its functions. They participate in various physiological jobs required from the brain to maintain and regulation its activities. Although these exert advantageous effects in normal conditions, if they are altered, can develop several brain damage. Next, please. Lipid-altered metabolism is considered as the hallmark of the major brain disorders. Lipid oxidation derivatives, mainly those that are not endogenously formed by enzymatic reactions, are strongly indicated to be part of potential harm. Dietary lipids, imbalance, oxidative stress, impaired autophagy, imbalanced gene expression are some of the major factors that can contribute to the alteration of this lipid metabolism. Click, please, Erin. Okay. Due to their pro-inflammatory, pro-oxidant, cytotoxic, and mutagenic effects, and apoptosis, lipid peroxidation, these are the major contribution to start a generation of chronic diseases or neurodegeneration. Next, please. Cholesterol is a key molecule. It's a multifunctional molecule, an important building block part of the cell membrane, participate in the bile acid degradation, is critical for placenta and the baby development, is important precursor of hormones and vitamins, and obviously is critical for the normal brain function, including neuronal repair, membrane remodeling, and plasticity. We will focus the importance of the cholesterol in brain ages. Next. Cholesterol is produced in situ in the brain, where its content exceeds more than any other in other organs. Since the blood-brain barrier prevents the uptake of lipoprotein from the circulation, the novel cholesterol is synthesized in the brain from the hydroxymethylglucuryl coenzyme A reductase. Cholesterol is required to form synapses in neuronal cells. Also, cholesterol facilitates the extension of axon survival on the neurons and also promotes the myelin formation. 70% of brain cholesterol is associated to myelin. Overall, it represents up to 25% of the total amount of cholesterol in the body. Brain cholesterol is essential to ensure cell membrane structure, neurotransmitter release, signal transductions, and synaptogenesis. Due to an unsaturation between carbon 5 and 6, cholesterol is susceptible to undergo oxidation, generating more than 100 oxidative species known as cholesterol oxidation products or oxisterols. These compounds can be generated by two major pathways. The first one is an enzymatic reaction through the cytochrome P450 family, and also no enzymatic reaction, generally starting by reactive oxygen species. Next, please. The cholesterol oxidation starts when this double bond in the carbon 5 and 6 break, generating several compounds very similar in the structure with the parent compound with different groups, could be a keto, diol, epoxide, etc. They are causing as the enzymatic and non-enzymatic reaction. Please click. And they have three phases as a regular lipid oxidation: initiation, propagation, when these species are formed, and termination. Next, please. Our laboratory has been explored, click please, and evaluated the major pathways of formation of these compounds. We have been identifying the enzymatic formation through the P450 family, the non-enzymatic formation through the reactive oxygen species, and also we have a third pathway that is one that comes from dietary intakes, being for us the most important to study. That's why we are super excited to combine on expertise in these compounds and the brain aging topic. Also, next please. We know that this oxysterol has been involved in the development of several chronic diseases. Some of them has been highly studied, such as cardiovascular diseases, hypercholesterolemia, diabetes, obesity, atherogenesis, and other has been raising in recent years, focuses in the brain aging, such as Alzheimer. Also, next please. Also, we have been tracking the profile and the amount of these compounds deposition in different tissues on how these depositions has been related with the generation of these chronic diseases. So as you can see, we have a database of literature reviewing the deposition of these compounds. And next, please. Most important, we know and we are able to profile and quantify each of the species, separating the pathway of formation. Next, please. Also, our laboratory has created a database of these compounds deposition in several tissues and biofluids. It's remarkable that we have the capability to distinguish among these three major paths of formation, and we have been found more than 20 different species in human placenta, breast milk, and amniotic fluid, blood, plasma, and different biofluids, as I mentioned before. Next, please. Okay, I will focus in the enzymatic oxysterols. In order to prevent cholesterol accumulation in the brain, cholesterol is oxidized by this P450 family. Cytochrome P450 are ubiquitous monoxygenase responsible for a variety of biochemical conversion, including steroid biosynthesis, drugs, and xenobiotic metabolism. There are membrane proteins differently distributed in the cells between endoplasmic reticulum and the mitochondria. Enzymatic oxysterols are mainly located in the side chains of the backbone of the structure. So we have positions 22, 24, 25, 26, and 27. The position 27 is a ternary carbon, which is converted by a non-enzymatic P450. Instead is a diuretic containing enzyme that converts it to the 25-hydroxycholesterol and is highly expressed in the immune system cells. Cholesterol is primarily converted by the CYP46A1 into 24-hydroxycholesterol, also named cerebrosterol. This component is able to cross the blood-brain barrier and reaches the peripheral circulation. We have another oxysterol is the 27-hydroxy produced by the CYP27A1 and then transformed into the 7-alpha-hydroxy-trioxo-4-cholestenoic acid, which also is able to cross the blood-brain barrier and reaches the liver when it is eliminated. Also, other oxysterols has been identified post-mortem. Some of them is the 7-ketocholesterol, 7-alpha-hydroxycholesterol, the isomers 5,6-epoxycholesterol, which these are not formed by enzymatic pathways. Mostly these are formed by reactive oxygens of a species. So for us, it's interesting to understand the relationship between the formation of these compounds and the brain aging. It has been not examined until now. Next, please. More in detail, the CYP7A is cholesterol 7-hydroxylase, is the rate-limiting enzyme and is the major pathway to eliminate cholesterol from the human body. The CYP27A1, the 27-hydroxylase, is an ubiquitous multifunctional mitochondrial monoxygenase, and it's expressed in the neurons, astrocytes, and the oligodendrocytes. Enzymatic activities of this 27-hydroxylation includes the bile acid intermediates in the liver and cholesterol in many intrahepatic tissues. We have also the CYP46A1 that convert to 24-hydroxy cholesterol, is a macrosomal monoxygenase expressed mainly in the brain and the astrocytes, where it's converted to the isomers 24 S and R hydroxycholesterol, which are able to remove the excess of cholesterol from the brain. So we identify these two major oxysterols as key components to understand the cholesterol homeostasis, where is an imbalance in the expression of these two molecules. Next, please.
2-minute warning, though.
Okay, it is well known that the hallmark for the pathology in the Alzheimer's disease is the aggregation of the beta amyloid peptides in the brain. However, we believe that the imbalance of these oxysterols compromise the cholesterol homeostasis, which is critical for the brain cells function and has been tightly linked with amyloid fibrillations. And as I mentioned before, a direct link between cholesterol and dementia progression has been now established in humans, and the molecular mechanism is still not elucidated. Please next. I will go quickly, Erin. So again, I will focus in these 24 and 27 oxysterols in order to understand if this imbalance might compromise the cholesterol homeostasis in the brain, and also if they can be potential biomarkers for an early detection of the stage of the disease. Next, please. Next, please. So our hypothesis is that the increasing of oxysterol concentrations in the brain might promote cellular damage, causing neuron dysfunction and degeneration, and could contribute to the neuroinflammation and amyloidogenesis. We have two major aims for this pilot study. Click, please. The first one is to correlate the systematic profile of these oxysterols, both enzymatic and non-enzymatic, and correlate with the progression of the dementia using a quantitative lipidomics. And also the second aim is obviously evaluate the expression of the cytochromes P450 that are those ones that generate these oxysterols using a transcriptome proteomic approach. Next, please. This is the potential mechanism. We are expecting that the production of the 24-hydroxycholesterol, it will be downregulated. And obviously, if this starts to be downregulated, less cholesterol in the brain can be removed. In the opposite, the 27-hydroxycholesterol will be accumulated in the brain. And with these two key components, we start compromising the cholesterol homeostasis. Click, please. Also, we know that there is an oxidative stress that generates no enzymatic oxysterols, but also there is a vicious cycle when oxidative stress promotes inflammation and inflammation promotes oxidative stress. So we are expecting that an increase in the inflammatory biomarkers such as cytokines will be generated. And obviously we are expecting that with this, the development of amyloid beta protein plaques will start in the different stage of the diseases. Next, please. So we have.
Are you almost finished? Do you have
yes,
many more slides?
Yes.
This is your last slide. Great, wonderful.
Thank you. We are using the Michigan Brain Bank biospecimens. Thank you to them. We have this cohort when we have highly likelihood Alzheimer's disease and low high likelihood Alzheimer's disease. We have 4 areas of the brain, and until now we have identified, please click, different oxysterols, 2 from an enzymatic origin, 3 from both enzymatic and non-enzymatic origin, and 7 from non-enzymatic origin. Click, please. It's remarkable and surprising for us that we confirmed that 27-hydroxycholesterol could be the potential biomarker to non-invasive detection in early stage of the disease, but also we have another potential biomarker that is the 5,6-epoxide, that is a non-enzymatic oxysterols. And again, we're very cautious because this is a pretty early stage of the study. We stopped it because of the COVID-19, and we are expecting to come back to confirm and expand these results when we are allowed to work in MSU. And the final slide will be obviously to thank my group, my collaborator, Dr. my collaborator, Dr. Carlo Barnard, for the Institute of Quantitative Health, my mentee committee, Dr. Paulson, Dr. Godreau, Dr. Morgan, and my students that has been helping me to perform this analysis. And I'm ready to take any questions. Thank you.
Thank you very much, Ilse. And I think, like so many of us, you're itching to get back in the laboratory to continue your studies. And I hope that at Michigan State that proceeds in a safe and an effective way, as it's starting to at the University of Michigan. For the sake of time, perhaps one question for Ilse, and then we'll have to move on if there are any questions.
Hi, this is Kevin Park from Central Michigan University, and I have a quick question. Hello?
Yes.
Yep, we can hear you.
Thank you for a nice talk. And actually, I was curious, with the aging, there is quite a bit of lipofuscin that does get deposited in aging brain. Any of the oxysterols that you are looking at, are they part of the lipofuscin that develops in the aging brain?
Say again, please.
With aging in the brain, lipofuscin deposits, and it's highly correlated with aging itself. The oxysterol species that you're examining in your study, are they components that get deposited in the lipofuscin in aging? I'm just kind of wondering.
There is no evidence until now of that relationship. They comes mostly from direct cholesterol, noble cholesterol. So until now, there is not so much information. That's why we are looking to first understand if there is a real connection between the different stages of the disease. And also our major target is to find if there are a potential biomarker of non-invasive tests, because obviously if we're able with these 2 or 3 oxysterols to confirm the early stage of the disease, it will be great.
Thank you, Ilse. And Kevin, thank you for participating. It's great to have our colleagues at Central Michigan also in the House. Folks, it is almost 11. We have 2 great rec training presentations to come. We have Courtney Polenik and Omar Ahmed. I highly recommend that you stay online for the next 2. And Courtney, you're next.
Thanks, Hank. Good morning, everyone, and thank you for joining us. Today, I'll talk about some recent projects on couples living with early stage dementia. Next slide, please. We know that most research on living with chronic conditions such as dementia focuses on individual characteristics and outcomes, such as the link between stress and depression. Next slide. We also know that people live in the context of their close relationships. Next slide. We typically don't consider the characteristics and outcomes of both individuals and close others. Next slide. Next slide, please. But health,
back.
Can you go back, please?
Yep.
Thank you. But health outcomes are often highly correlated within close relationships, and the characteristics of close others are often linked to individual outcomes over and above individual characteristics. So considering these mutual influences may provide additional information about factors that impact health outcomes and could enhance clinical care. Next slide, please. First, I'll talk a little about some of my previous research in the context of chronic illness and dementia. Looking at the next slide, most of my work has examined mutual influences among older couples. Spouses are a major source of health-related influence and support, so they can play a major role in shaping one another's mental and physical health. Next slide. Using data from the Health and Retirement Study, my colleagues and I have found that husbands' greater pain intensity is associated with wives' increased depressive symptoms over an 8-year period. And this might be because wives generally provide more health-related support to their spouses, and this may have a negative impact on their own well-being over time. We also found that husbands report higher depressive symptoms over time when they have health conditions that require different self-management activities from their wives' conditions, or what we call discordant conditions. And this may be because men often depend on their partners for health-related support more so than women, and the quality and quantity of the support might be lower when wives have self-management goals for their conditions that differ from those of their husbands. So as a whole, this research suggests that older spouses affect one another's mental and physical health trajectories. Next slide. Next slide, please. I've also done some work focused on dementia caregiving using data from the National Health and Aging Trend Study and National Study of Caregiving. And for example, one study found that caregivers report more emotional difficulties from caregiving when persons living with dementia have comorbid diabetes or osteoporosis. In another study, we found that caregivers report greater rewards or benefits from caregiving when persons living with dementia perceive greater purpose in life. And this is even after accounting for caregivers' own perceptions of their own purpose in life. And these studies indicate that both health-related and psychosocial characteristics of persons living with dementia have important implications for caregiver outcomes. Next slide. Next, I'll talk briefly about two current projects that focus on couple relationships. Next slide. The first project is the Couples Wellbeing and Everyday Life Study, or the CWEL study. Next slide, please. So the overall goal of this study is to understand how daily stress, self-care behaviors such as sleep and physical activity, and well-being are linked among couples living with early stage dementia. Next slide. We'll recruit 50 couples in which one partner has either a diagnosis of dementia in the past 5 years and symptoms consistent with early stage dementia, or symptoms consistent with early stage dementia but no formal diagnosis. And we're using spouse reports on the functional assessment screening test with scores of either 3 or 4 to indicate mild or early dementia. Next slide, please. This study includes baseline interviews and daily phone interviews over a week period for both partners. And we're also collecting saliva samples 4 times a day across 4 days. So we'll be able to examine both subjective well-being and also biological reactivity to stress. We started our recruitment for this study last August, and we're currently paused due to COVID, but we're working on some ways to modify our procedures to eliminate in-person contact with participants and to safely resume our saliva collection. Okay, so looking at a simplified model for this study, we're considering both partners' reports of daily stressors and their associations with daily mood and biomeasures of stress, specifically alpha amylase and cortisol, which play important roles in biological stress reactivity. Next slide. Okay, next slide, please. We'll also consider how each individual affects their own outcomes as well as their partner's outcomes. And we'll assess both partners' background characteristics and psychosocial resources such as coping strategies and self-efficacy. Next slide. As well as how these characteristics might affect how daily stressors are linked to outcomes within the couple. Finally, we'll consider co-regulation in partners' daily mood and stress reactivity. Next slide, please. And next slide. And the potential moderating roles of background characteristics and psychosocial resources. Next slide, please. The next project is the Self-Management, Health, and Resilience in Partners study, or the SHARP study. Next slide. The goal of this study is to understand the experiences of couples living with multiple chronic conditions. And because we were just about to start data collection when the pandemic began, we decided to include questions about COVID-related experiences before and since the pandemic. Next slide. We just started our recruitment of 40 couples in which both partners have 2 or more chronic conditions and at least one partner is age 50 or older. And we're interested also in considering dementia as a condition that may impact both partners' self-management. So we'll include a subsample with one partner having early stage dementia. The study will involve one phone interview with each partner, and this will include background and health questions, along with a semi-structured interview focused on their individual and joint management of multiple chronic conditions. Okay, next slide, please. And to participate in this study, both partners need to have at least one of these medical index conditions, and these are all strongly associated with morbidity and mortality. Next slide, please. So thinking about practical implications of this work, this line of research has shown that care dyad members mutually shape one another's health and well-being, and understanding these mutual influences may inform more targeted and personalized clinical care. This work may also help to identify individuals who are most in need of intervention, as well as those who may benefit from a couple or family-based approach to maintain their mental and physical health. Next slide, please. I'd like to acknowledge my major collaborators and study team members on these projects, as well as my support from the NIA and the MADRC. And I welcome your comments or questions.
Thank you very much, Courtney. And I'm really pleased to see that you have incorporated questions about COVID-19. This is going to be affecting all of us, but particularly those who are older, and whether they've been infected or not, whether they've had someone in the family affected. So I really appreciate the fact that you're taking that into account when you do these very important studies. Questions for Courtney?
Hi, my name is Deanna Bird and I'm at Wayne State University. I had a quick question for you. I noticed in some of the conditions that you had for the chronic conditions, you didn't have anything around hypertension. So I just wanted to know if that was something that you just excluded. And also in terms of race, was there any specific racial demographic that you were targeting? Obviously, we know, particularly with COVID-19, having a greater impact on communities of color. So I just wanted to see if any of your work was addressing any of those aspects.
Yeah, so first for hypertension, we are still including people with hypertension, but the medical index conditions, Erin, if you could actually go back to that slide just so we can have a look at that. So these are conditions that we just want to make sure we're sort of having some kind of consistency in our sample in terms of part of the picture of chronic illness that people are dealing with, but we are certainly also still including hypertension as one of the conditions of interest. With regard to your second question about racial disparities, we are definitely trying to include as many participants who are Black or African American to look at disparities, because certainly with COVID, as you mentioned, that is something that's important.
Other questions? Well, thank you, Courtney. We really appreciate it. And I look forward to working with you further on various issues that relate to wellness in the center of the next several years. Last but not least, we have Omar Ahmed will be presenting, and he's from the University of Michigan. I do want to say before we get to Omar that I believe that Renee not tweeted out, but she sent out to all of you the evaluation form. I really encourage you to fill out the evaluation form. That's how we know what we do right and what we don't do right. And particularly in this new era of remote conferencing, we have a lot to learn. I've really enjoyed these presentations and have felt them to be very, very informative for me and for our audience, but you may feel differently. So please do let us know your thoughts and your comments for making this even better down the road. Okay, Omar, you ready to go?
Yes, I am. Thanks, Hank. So I'm going to be talking about a brain region called the Retrospinal Cortex, which is one of the first regions to show dysfunctional activity in patients with early stage Alzheimer's disease. Next slide, please. 93% of Alzheimer's patients tend to get lost. They have spatial disorientation. And this isn't due to traditional memory impairments. Getting lost in this way and having spatial disorientation reflects the inability to link the outside world with your internal navigational goals. And that's why even in familiar settings, you can get lost. This is also seen in Parkinson's patients. Again, 90+% of Parkinson's patients have it. Next two slides, please. One more. And I think this quote, which is from a Parkinson's patient, but applies just as well to Alzheimer's patients, sums up the phenomenon. I used to walk alone in the wood, fog or no fog, but when the symptoms of Parkinson's appeared, I noticed I could no longer orient myself. And in case of fog, I got lost. Now I am too disabled to get lost anymore, which is why spatial disorientation is underdiagnosed in advanced stage Parkinson's disease. But it is a phenomenon. It accompanies both Alzheimer's and Parkinson's disease, and it needs to be understood. Next slide, please. Here's another patient. He was referred to our department 2 weeks before while driving his car. He suddenly realized he was unable to recognize the streets along the way home. He got lost and had to call passersby to help. Since then, he's been severely disoriented in space. Next slide. One more, essentially the same thing. Consequently, he could not determine which direction to proceed to go home. Next slide. So neither of these patients is an Alzheimer's or Parkinson's disease patient. Next slide. Both of them had damage to their retrospinal cortex, leading to essentially the same phenotype that is seen in patients with Alzheimer's disease, an inability to orient yourself in space and find your way home, essentially. Next slide. This is what it kind of manifests itself as. Patients with retrospinal damage essentially cannot draw their neighborhood with as much detail as their sons can in this example, and relationships between neighboring landmarks are impaired. So in this case, the home and library and parking are all offset from each other as they should be. Next slide. And the retrospinal cortex is also critical for what we think of as traditional memory formation. Here, a patient who had retrospinal damage essentially had major retrograde amnesia and anterograde amnesia as well. Next slide. And we're focused essentially on understanding the neural circuitry of the retrospinal cortex, how it contributes to spatial orientation, and how this precise circuitry is impaired in early stage Alzheimer's disease mouse models and rat models. One of the problems with wanting to understand the role of the retrospinal cortex in Alzheimer's is that if you wanted to understand the hippocampus, you would go to PubMed, you'd get 15,000 results if you search for hippocampus Alzheimer's. If you do the same for retrospinal Alzheimer's, you get about 100. And there isn't much of a background literature about retrospinal cells, oscillations, brain rhythms, circuits to actually build upon. So what we've had to do is basically lay those foundational principles down for the retrospinal cortex, and then understand how those foundational principles change in Alzheimer's. Next slide, please. There's where the retrospinal cortex is located in the human brain. It sits behind the splenium of the corpus callosum, which is why it's called retrospinal. It has two major subdivisions, granular and dysgranular. We focus on the granular, which we think is the more important subdivision for Alzheimer's. Next slide, please. And that's what it looks like in the mouse. It sits very medially and essentially runs along the entire long axis. Next slide. These are the kind of tools we use: virtual reality coupled with behavioral electrophysiology, 2-photon imaging, and computational modeling along with slice work. So we try to bring all the cutting edge tools to the understanding of retrospinal circuits in Alzheimer's. Next slide. So I'm going to tell you a little bit about retrospinal rhythms and how they change in Alzheimer's, and our preliminary results indicating that cholinergic impairments may underlie the impairments we see in the retrospinal cortex in Alzheimer's disease. Next slide. All right, next slide. So we began by doing a survey of brain oscillations, including the retrospinal cortex, hippocampus, posterior parietal cortex, and a few other neighboring areas. And what we found was, next slide, by recording essentially for really long periods of time, we characterized their sleep oscillations in non-REM sleep, REM sleep, as well as their awake behavioral oscillations. Next slide. And here's really the key thing that we kind of observed. So if you see up above in the hippocampus, during non-REM sleep, you get a lot of those red streaks, and those are hippocampal ripples that are well known. They play a major role in learning and memory, and specifically memory consolidation. So hippocampal ripples are critical, and you see them in non-REM sleep. But in the retrospinal cortex, you barely see any ripples, but instead during REM sleep, you get this other unique oscillation coming up that isn't seen in the hippocampus. It's almost as if these high frequency oscillations are alternating between non-REM and REM sleep between the hippocampus and retrospinal cortex. And they are interconnected structures, which suggests something really interesting is happening during REM sleep in terms of memory consolidation, even in the retrospinal cortex. Next slide, please. And these oscillations are significantly more powerful in the retrospinal cortex than any other brain region studied. Like it's not even close. Next slide, please. And what did they look like in the raw EEG signal, raw local field potential signal? Next slide. That's essentially what they look like. If you focus your attention in the top right corner there, that is what's called a single theta cycle. And if you see those little teeth-like things, those are the fast oscillations we're talking about. They occur at the peak of theta oscillations, and you can see that at the bottom row there with that sharp red demarcation right there. So these things are happening selectively during theta oscillations in REM sleep and awake behavior, and they're strongest in retrospinal cortex. Next slide, please. because of how they look, we needed a new name for them. They're not like any other oscillation that's been described in other brain regions. So splines are the teeth that sit atop mechanical gears and essentially interlock. And so we decided to call them splines. So these are retrosplenial splines, fast oscillations that essentially are a hallmark of retrosplenial activity. Next slide, please. And we found that retrosplenial splines demarcate high activity REM frames. So what does that mean? Well, whenever during REM sleep, the brains of these mice and rats become more active, that's when these spline oscillations kick in. When I gave this a similar talk to a sleep audience, they described the gray dotted lines that you see on your screen right now during one REM sleep epoch as reminding them of the probability of occurrence of dreams in humans when in older studies where they used to wake patients up during a single REM epoch. So we're not saying they're dreams, but they do seem to, and we don't know exactly how REM dreams look like in mice and rats, but they do seem to demarcate pronounced high activity periods during REM sleep. So they're important. Next slide, please. We've characterized, I won't spend too much time on this, but we've characterized the exact cell type specific nature of these oscillations, and it involves these fast spiking neurons in the retrospinal cortex. Next slide. Next slide, I'll just skip over this part. And what we found is basically it needs an FS-FS connectivity to those aficionados out there who study fast spiking cells, and those are necessary to generate splines. Next slide. And we made computational models that can essentially reproduce, if you look at D and E here, we can reproduce the spline dynamics that are seen in the in vivo data using a simple computational model. So we're fairly confident we understand how this circuit works. Next slide. So, but what happens to these? So we've described a phenomenon in the retrospinal cortex that is central to retrospinal cortical function during sleep at least, and as well, I'll show you also during awake states. But what happens to these oscillations during REM sleep in a rat model of Alzheimer's disease? The TG344AD model is what we started with. Next slide. And it's a pretty clear decrease that you find that these spline oscillations during REM sleep. So the rats don't have to do anything here. These are fairly young rats, 3 months of age. They're just sitting there, they're sleeping. And even during sleep, even without having to do any behavioral tasks, their spline oscillations are significantly reduced compared to wild types. Next slide. And because there are so many more mouse models compared to rat models, we then established, we first asked, do splines occur in mice? And if so, how do they change in mice? Next slide. So this is our behavioral setup. We can rotate now, we can head fix, we can record, we can do all kinds of things in this particular head fix setting. And then the mouse is also free to walk around afterwards or go to sleep, as you can see in D. Next slide. So by doing this, what we found is these spline oscillations do occur during awake behavior. Not only that, they actually get stronger and more precise as the mice run faster. So they're navigationally important. The retrospinal cortex is navigationally important. These oscillations are navigationally important. And that red trace there in E shows you how these splines are essentially going up as a function of speed. So this was in wild type mice, but what happens in a 5x FAD mouse model of Alzheimer's disease? Next slide. And this is really, I think, one of the key results here. So if you compare E and H, in E, in wild type animals, splines are essentially going up with speed. But in 5x FAD mice, splines are actually decreasing with speed. And as you run faster, typically that's supposed to lead to a stronger signal in the retrospinal cortex, but that isn't happening. So when you're actually moving around, when these mice are moving around, the spline oscillations that help to coordinate circuit activity are impaired as they move at faster speeds, which may correlate to the spatial disorientation that is seen in patients. Next slide. So last few slides here, what I want to kind of try to get at is what our lab is now trying to do to understand why the retrospinal cortex is altered in mouse models of Alzheimer's disease and rat models as well. Next slide. So if you look at amyloid plaque distribution in both rats and mice, and if you look at the retrospinal cortex, hippocampus, and entorhinal cortex all at the same time, at the same age, you see something pretty interesting. In the hippocampus and the entorhinal cortex, which are again the traditionally studied brain regions for understanding spatial navigation in Alzheimer's disease, at this age point, which was 12 months, you see lots of plaque deposits in both the entorhinal cortex and hippocampus. This is why people have been drawn to it, even though the retrospinal cortex in humans shows early dysfunction. But if you look at the retrospinal cortex, you don't see many plaques. That's not the reason. This is not the underlying reason for the differences we are seeing. It's preceding plaque formation in the retrospinal cortex. Next slide. And cholinergic projections are one key source of external input to the retrospinal cortex. So we started to document exactly how the cholinergic inputs to the retrospinal cortex look. I don't need to mention to this audience how important cholinergic hypotheses are in the Alzheimer's field. So what do cholinergic projections to the retrospinal cortex look like? Next slide. And you can see over here, this is marking the vesicular cholinergic transporters, essentially the synaptic terminals. And you can see right in the middle of your screen there, there's a dense projection right to layer 1 of the retrospinal granular cortex, the very region we're studying. It's as strong, and if not maybe denser than what you see in the hippocampus. Next slide. Next slide. This is a beautiful little zoom in here. Again, you can see this beautiful laminar pattern in layer 1A and layer 4 that actually, for the aficionados, overlaps beautifully with thalamic input to this brain region as well. So very precise modulation of these circuits would be predicted based on this lamination. Next slide. So that is all wild type, but in 5XFAD, that dense band in layer 1 is decreased and way less dense as early as 3 months of age, suggesting that these terminals are, fewer of these terminals are projecting here, consistent with the cholinergic hypothesis of basal forebrain cholinergic cells failing early on in Alzheimer's. Next slide. And this is pretty much my last data slide. So what we're doing, and this is work by Isabella in the lab, what we found is that these retrospinal cells, pyramidal cells in layers 3 and 5, are very sensitive to cholinergic agonists. They respond with this persistent activity in the left 2 columns. When you bathe them in cholinergic agonists, you get pronounced persistent activity. This has been seen before in the entorhinal cortex and other brain regions as well. And it's thought to underlie spatial memory and working memory. It's basically important for spatial navigation is what's thought. But we are not seeing this persistent activity in response to cholinergic agonists at all yet in 5XFAD mice as early as 3 months of age. So it's not just that the cholinergic inputs are gone, and we've shown that this is atropine sensitive, so it's muscarinic, but there aren't the muscarinic receptor-induced persistent activity is absent. And this could also explain why muscarinic agonists haven't been as successful as one might think they should be. Essentially, we think either they are knocked down early on, or the chain reactions from the GQ protein are essentially dysfunctional very early on in Alzheimer's disease. And now we're trying to build a cell type specific map to understand exactly in what cells and at what time point this muscarinic induced persistent activity is knocked out, so that then we can try to restore that and see if we can improve spatial orientation in mouse models of Alzheimer's. Next slide. That's the summary. It basically repeats everything I just said. Next slide. A lot of people to thank, especially MADRC for the pilot grant, for all the advice. I also want to thank Jeff Murphy, Shannon Moore, and Rachel Parent in Jeff's lab for providing us with the 5XFAD mice, without which we couldn't have gotten started, and looking forward to collaborating with all of you over the coming years. Thanks.
Thank you, Omar. I don't think you use the word aficionado. I don't think one needs to be an aficionado to see how fascinating and potentially very important this is. So I really appreciate you bringing home our symposium with this really rock star talk. I really enjoyed that. I could have questions, but I am sure there are questions from the audience.
This is Kevin Park again from Central Michigan University, and I have a question. And your work is quite fascinating. You use 5XFAD and 433. Those are overexpressors. Do you have any plans to study the APP on the left, the knock-ins from maybe cyto group or other knock-in animals in your study?
That's our next plan. It's we started with one rat and one mouse model to kind of essentially see if this phenotype was consistent, see if most of our observations were consistent. But now we want to extend it to other models because essentially we agree that there is no one perfect model here, and those are good candidates for the next step. Agreed.
And would you say that there is a maybe correlation between retrosplenial function and maybe tau? Because again, tau dysfunction is more correlated with cognitive dysfunction than amyloid beta deposits. In your study, you're showing significant defect in these APP models. Their tau dysfunction, their tau pathology is quite mild in these animals. Do you have any maybe insight or thoughts?
I think we're kind of stepping away from both tau and amyloid beta here, and just looking at cholinergic inputs, because one of the things about the amyloid deposits, for example, in the retrospinal cortex is one old paper that says that exactly where the cholinergic terminals are landing, those layer 1 and layer 4 spots I just showed you, that's where eventually at much older ages you will start to see amyloid beta deposits. So, what is the primary trigger for what happens subsequently? We believe it's cholinergic downregulation and subsequent dysfunction between the circuit activities. And that's what we're pursuing. And we want to see if this is happening as early as 1 month of age in these mice models, as well as the ones you mentioned earlier. So we want to kind of shift the conversation a little bit to just looking at cell type specific cholinergic modulation first, and then come back to understanding how tau and amyloid beta subsequently play a role.
I think it's great that you're looking at Alzheimer's. I think it's really important, and tau is a relevant issue here. Clearly, cholinergic circuitry is impaired significantly and can be modulated significantly in Lewy body disorders as well. And that would be, especially when you think about sort of more posterior cortical involvement in disease, think about things such as REM sleep behavior disorder, that the Lewy body disorders are directly up this alley as well. So there's an area of further investigation for you. I want to make a comment. The rec trainees that we talk about and the pilot awards we give are to help younger investigators. But I'm looking at your list here and I see Ellen Brennan is one of your students, and she gave probably one of the most exceptional lectures I've seen in the neuroscience program by the graduate students. So clearly the mentoring is occurring in your laboratory with graduate students as well. So we appreciate that. Other questions for Omar?
So this is Jeff Murphy from University of Michigan. Really nice talk, Omar. Just spectacular data set. You should be happy with your students. They're really doing great.
They're an awesome set of students. I didn't mention them enough here, but really, they're all doing a great job.
So I don't want to get too far down into the minutia, but I noticed in the current clamp recordings, the wild type animals seem to have a prominent ADP.
Exactly.
And the 5X SADs do not. Is that just a artifact of this representative sample that you showed, or is that really?
The ADP is missing in the 5XFAD. So the ADP is actually, I didn't want to get into that, but you're absolutely right. The ADP itself is gone, and hence any persistent activity that could accompany the slow ADP is also gone. You're absolutely right.
Yeah, we should talk about that offline sometime. That's really
I would love to pick your brain on that question. That would be fun.
Well, I'm sure people will have lots of comments and questions for Omar. I'm looking forward to discussing with him offline later. And we have a lobster to bring us home. That's wonderful. I got to say, I couldn't be prouder than what I've heard of the people who are interacting with the center. Such a range of expertise and explorations that are all relevant to cognitive impairment, wellness, dementia. It's what we're here for. And those of you who are listening in, who want to participate, we'd love to have you join us. I will encourage you, please, to go to that link that Aaron sent and that I think Renee also sent to evaluate today's conference. Stay in touch with us. Aaron, any final comments you want to make?
Just a couple admin things here on this slide. I've had a couple people reaching out interested in contacting our presenters. Please don't hesitate to contact me. I'll be sending a follow-up email here, so you'll have my email address at the top of your inbox, but it's also on this slide here. So feel free to contact me and I can get you the direct contact information for each of our presenters if you would like to contact them and you do not know it. Again, just the event eval, as Hank mentioned, the recording of this event will be on YouTube probably early next week. And the link to that is right here. I'll also send it in my email follow-up here. Again, save the date for 2021. I know summer is always a busy time, so get it on your calendar if you wish, and then just some ways to stay in contact with us if you want to know of other upcoming events or other opportunities. You can sign up for our monthly e-newsletters. That link is here, as well as our social media on the right-hand side. So I would love to stay in touch. Thanks, everybody.
Well, let me just say thank you, Erin and Renee and others who helped put this together. Thanks to all our presenters. And thank you who listened today. Be healthy, stay safe, and as you get back in the laboratory or back to seeing participants clinically, we appreciate what you do. Bye-bye.
Everybody be safe.